I'm not, it's not that bad. I don't think it's that. Okay, just you have to pick up like major concepts from each chapter. And some chapters like this, past few ones, had a lot of ideas. Okay, chromosomes. So, chromosomes, basis of inheritance. Mendel hereditary factors were purely abstract when first proposed. So when Mendel talked about those ideas at the beginning, it was all abstract. It was not, as I said, he referred to the genes as the elemente, and but now we know they're genes. Today we can show that the factors genes are located on chromosomes. The location of a particular gene can be seen by tagging isolated chromosomes with the fluorescent dye that highlights the gene. Here it is. They highlighted this is a chromosome, as you all know, and right here they tagged it, and you can see that gene on that chromosome. That's pretty much what it is. You have one gene here, one allele, one allele, one allele here, one allele here, one allele here, one. So the chromosome is full of genes. We already talked about that. You already know that. Okay, and then in the past, we talked about this independent assortment of independent orientation of the chromosomes. This is your Mendel independent assortment. You remember that? We talked about this. This is what Mendel proposed, and these genes are making the plants at the end of the plants. The ratio is 9 to 3 to 3 to what? I'm not going to go over that, but it's actually the orientation of the chromosomes. And the genes on the chromosomes make the plants, 9 of them round yellow, 3 of them round green, 3 of them wrinkled yellow, and 1 of them green wrinkled. Okay, it's this just one. So this is very important. This PowerPoints, I hope you sit down and look at it and understand it. And after that, I hope you can recall the information. Okay, Morgan's choice of experimental organism. Morgan was a scientist who, after Mendel, he came up with a lot of nice findings. Whatever we know of genetics today, first was Mendel, and then after that was Morgan. So what Morgan chose, study of the Drosophila melanogaster. It's a fruit fly. It's a type of fruit fly. Several characteristics made fruit fly a convenient organisms for genetic studies. Today, already talked about that. Human is not a good subject for genetic studies. We live long, 10 years, and then we cannot mate. We are mating randomly, and then our offspring, we have 1 or 2 offsprings. That doesn't work. That does not have a good indication of genetic studies. But they produce many offsprings, the fruit fly, and a generation can be bred 2 weeks. And they have only 4 pairs of chromosomes. They do not have, like us, 23 pairs. They only have 4 pairs of chromosomes. Nice, easy to study. Very easy, very nice to study. Easy to grow in the laboratory. Very easy to grow in the lab. So Morgan noted wild type or normal one. Here we go, finally I'm claiming that what is wild is normal phenotype that were common in the fruit fly population. Traits alternative to the wild types are called mutant phenotypes. Here they are. That's a normal type. That's a normal eye. So he bred these fruit flies. And then after he bred them, he looked at them on the dissecting scopes, see which one have red eye, which one have white eye. The red eye was the wild type. Wild type, it means the one that is normal. That's what it's supposed to be. Okay, the white eye are the eyes that are the mutant ones. Okay, that's what Morgan studied. Then correlation behavior of a gene's alleles with behavior of the chromosomes pairs. In one experiment, Morgan made it male. Now we're talking about male and female. Before, during Mendel, male and female did not mean anything. But now, with Morgan, he came up, and then of course in fruit flies, you know which one is male, which one is female. The sexes are separate. They are not monoecious. The pea plants that Mendel used, they were monoecious. Monoecious, it means both sexes are in one organism, hermaphroditic. You all know the term hermaphrodite, hermaphroditic. It means both sexes are in one species. But in fruit flies, that's not the case. You have testes in one organism and ovary in another organism. Two separate organisms. These animals are like us, we humans are dioecious. We refer to us as dioecious. So, he said male flies with white eye, mutant, the male are white, that would be the male. And then with the female flies with the red eye, so that would be female. Male, female, and he crossed them together. The F1 generation all had red eye. So red was dominant over white. And then what happens? F2 generation, he saw what Mendel saw, 3-to-1 ratio. Okay, 3-to-1 red to white eye ratio, but only males had white eye. Only male ones had white eye. The female ones, they were all red. Morgan determined that the white eye mutant alleles must be located on the X chromosome. X chromosome is, as you know, it's a sex chromosome. Okay, Morgan's findings supported that the chromosomes theory of inheritance. Here they are, the X chromosome, whatever you read in the last PowerPoints, here is summing it up for you guys. I'll let you take a look at it and go over it. Here is the X chromosome, here is the Y chromosomes in karyotyping. Of course, you know, we humans have 22 pairs of autosomes, 22 pairs of autosomes, and one X chromosome and one Y chromosome. So that one is a male, XX is a female. So for us human, we use X and Y. For poultry, chicken, they use ZW. Okay. For grasshopper, the female, it is XX, and the male is just one X. The male in grasshopper. And then in the bees that you know something about, some of you are diploid, that would be the female, and the haploid male have 16 chromosomes. Okay, so those are some of the haplodiploid system, and then of course this is the X0 system. We human are XY system and ZW system. Short segments of the Y chromosomes are homologous with X, allowing the two to behave like homologous during meiosis in males. A gene on the Y chromosomes called SRY gene. Why they call it SRY gene? It means sex determining region. Sex determining SRY, S for sex, region, I'm sorry, yes, region and Y. So is responsible for development of the testes in an embryo. So the reason that we are males because we have this gene and the females do not have the SRY gene. That's pretty much it. Okay, just one gene determines the sex of the organism. A gene that is located on either sex chromosomes is called a sex-linked gene. That's what we are going to talk about next. Gene on the Y chromosomes are called Y-linked genes, and there are a few of these. Genes on the X chromosomes are called X-linked genes. X-linked genes follow specific patterns of inheritance. For recessive X-linked traits to be expressed, a female needs 2 copies of the alleles homozygous, and male needs only 1 copy of, that's colorblindness. Homozygous, I'm going to talk about that. And then X-linked recessive disorders are much more common in males than in females here. Okay, I'll let you look at that one at home, but I want to go over it on the board. This is what I was talking about. The highlight of this chapter is just this one PowerPoint. But again, there are a few other good information in there. It's just this one page. Pay attention. Hopefully, you would know. So you know Mary is XX, is that right? And Adam is XY. Yes, you all agree? What are the possibility of the sperm from Adam? Huh? Hello? Anybody home? What is the possibility of sperm? X is one possibility of sperm. The other possibility of the sperm is Y. What is the possibility of the egg from Mary? What is the other possibility? X. So, they get married and they have kids. What is that one? It's a female. Girl, big girl. What is that one? It's a male, it's a boy. And what is this one? Female. And what is that one? What is the ratio? Huh? What is the ratio? 1 to 1. But is the population of the earth 1 to 1? Very close to it. There are more males on the planet earth than females, a little bit more, based on your textbook. I don't believe it, but I don't go around and to me it seems like there are more females than male, but that's not true. I guess I'm getting them mixed up now. One of them is a little bit more, but the ratio is still 1 to 1. The ratio of the people on planet Earth is to 1 to 1. Okay, let's talk about a gene that is Mary has a gene. Let's talk about it that causes car blindness. I'm going to indicate it as a C, and I'm going to put it on the X chromosome. That X chromosome. Is Mary colorblind? Is Mary colorblind? No. Mary is a carrier. I said it. Mary is a carrier, and she's not colorblind, but she carries the gene. Right? Am I making some sense, everybody, so far? So when she's giving off her eggs, let's say this one has the gene, that one doesn't. Am I making some sense, everybody, so far? So she has the gene on this chromosome. When she goes through meiosis, she makes eggs. So tell me about this one. Is this girl a carrier or a normal girl? Normal girl. She's a normal girl. How about that boy? Is she a carrier or a normal boy? Normal. Okay, how about that girl? What is she? She's a carrier, but she's normal. Carrier, but normal. You all know what I'm talking about. And here is the gene on, right? She got this from mama, from Mary, and she got this X from Adam. You all follow what I'm talking about? Am I making some sense? Now, how about this one? This boy is what? Is color blind. That boy is colorblind. If they have all of these children, 3 of them are normal, 1 of them is colorblind. Okay? Then I'm continuing on this diagram. Then let's say he gets married, this boy. He gets married with a girl that is also a carrier. Like this, and of course that one, right? You all follow what I'm talking about? He is colorblind. She is carrier, but normal. He doesn't know that he is colorblind, right? That she's a carrier, not colorblind. She knows that he is colorblind, right? You all know what I'm talking about? Yeah. They go on a date, say, "Oh, honey, did you know I'm colorblind?" Really? How does that feel like? What is that like? What color do you see? What don't you see? Oh, I still love you, I'll marry you. Right? Am I making some sense, everybody? And he has no clue she's a carrier. She's normal. Can you see this, honey? Oh yeah, it is red. Oh, okay, I'll marry you then. He tells her, can you see this? I say, no, I can. Well, let's say this one is John. John says, no, I see it, but I see it as a gray. And then this one, what is her name? Mary, Lisa. Lisa, can you see this? Yeah, of course it's red. Oh, okay, I'll marry you then. Now, are you listening? Are you ready? They have children. They get married. They have children. And then, of course, in his sperm, these are the possibility of sperm, right? Right? And what are the possibility of the egg? These are possibility of the egg. Do that make sense? They have baby, they're married, they have a child, and that child is a girl, and what is she? Tell me what her status. She has a carrier, but she's normal. She sees this as what? Red. She sees it, the baby sees this as a red. Okay. Now they have another child. That child is a boy, right? Am I making some sense? What is that one? What is that one? He's normal. He's normal. He sees this one as red. Okay. Now they have a daughter. Most textbooks fail to mention this. Your textbook did mention it. They have a daughter, and that daughter is what? Colorblind. That daughter sees this as gray. That daughter sees this as a gray. They have another child. What is it? What is that child? Huh? Colorblind. That child sees this as gray. And then John says, hey, I didn't sign up for this. I didn't want two colorblind child. You lied to me. I want divorce. They hire Amir as a consultant, give him $1 million. And then I have to sit down and go through a semester of biology and explain it to John. John, you're wrong. Lisa is right. Right? Isn't it? Most of us get married. After a few years, we say, oh, I didn't sign up for this. I still recommend it to everybody. Everybody should get married. Even though you don't sign up for it, you should get married. But anyway, so this is pretty much what that is. What I wrote on the board. Okay, so it is possible to have a very rare to have a colorblind female, but it is possible. Most colorblind people are male, as you know why. Okay, most colorblind people are male, but it is possible to have colorblind female. It is possible. So, some disorders caused by recessive alleles on the X chromosomes in humans, colorblindness is one of them. Muscular dystrophy is another one. Hemophilia is another one. Bleeders, you know, Bleeders disease. That's what hemophilia is. Recombination of unlike genes, independent assortment of chromosomes, offsprings with phenotypes matching one of the parental phenotype are called parental types. Offsprings with non-parental phenotype, new combination traits are called recombinant types or recombinant. I'll show you a 50% frequency of recombinant is observed any two genes on different chromosomes. For example, I do not have one. This sex link is a bad example of this one. Here we go. This is very important that you have to, that's what happened to Mendel, and that's what Mendel saw. So one parent is capital Y, small y, capital R, small r, right? That's one genotype of one parent. The other parent is small y, small y, small r, small r. Do you guys see that? When you cross them together, this type of seed with that type of seed, what you're going to get? Does this look like the genotype of this one? Does it look like a parent?
Yeah.
Yes. How about that one? Does it look like of the parent?
No. Oh, yeah.
Small y, small y, small r, small r. Does it look like that parent?
Yeah.
Yes. How about that one? Does this one look like any of the parent?
No.
That's what this is called, a recombinant. This is called the recombinant chromosome. It's the same as the last one that recombinant offsprings. Right here, recombinants. That's what these are. How about that one? Does this one look like any of the parents? No. So these two are recombinant. These are parental type offsprings. And then I will ask that during the exam, and you say, Amir, what do you mean by that? Right here. That's the PowerPoint. Recombinant, it means these two, look at their capital Y, small r, small r, does not look like any of the parents. The genotype, I'm talking about the genotype. Same as that one. Small I, small y, capital R, small r. None of them. The whole entire, I'm talking about the whole entire genome in this case. Okay? So that's what these are called. It doesn't make sense. The example of the X chromosome is not a good example. What we have here. It's not good. Even though this is, you can call this as a recombinant one too. Yeah, sure, why not? This doesn't look like any of the parents. This is a recombinant one. Why not? That's a recombinant one. Does not look like any of the parents. Same as, no, that's it. Same as that one. That one is a recombinant one too, isn't it? So we do have some examples in there. Morgan discovered that genes can be linked, but the linkage was incomplete because some recombinant phenotypes were observed. Recombinant phenotypes were observed. He proposed that some process must occasionally break the physical connection between the genes of same chromosomes. That mechanism was crossing over homologous chromosomes, which we talked about that. And then, of course, he talked about genetic map. A few people after him, they talked about genetic map, where the location of the chromosome, where the location of the genes are on the chromosomes. That's called genetic mapping. They do that for like people who want to go in for breast cancer, see if their family had breast cancer, they have breast cancer, they map their genes, and then based on that, they find out that's what the mapping is. They find out if the person has it. What else? Alternations of chromosomes numbers or structures cause some genetic disorders. Large-scale chromosomes alternation in humans and other mammals often lead to spontaneous abortion, miscarriage, and plants tolerate such genetic better than human. For example, if you human are XX, 3 triple X and then Y, sometimes the baby will die. Okay, that cannot happen. We cannot have triple X and then Y. It has to be double X and Y is okay. Usually they survive, but triple X, Y usually they die. Things like that. Abnormal chromosomes numbers. It is non-disjunction. Pairs of homologous chromosomes do not separate normally during meiosis. As a result, one gamete receives 2 of the same types of chromosomes, and another gamete receives no copy. Right here. So that is abnormal. Some of these, remember, I talked about this. This is called a video showing non-disjunctional chromosomes. Aneuploidy, and in front of a word means what, guys? Without. Euploidy means without correct results. Euploidy results from fertilization of the gametes in which non-disjunction occurred. Offsprings with this condition have an abnormal number of particular chromosomes. Monosomic zygote has one copy of particular chromosomes. A trisomic zygote has 3 copies of a particular chromosomes. Polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes. Triploidy has 3 sets. Tetraploidy has 4 sets of chromosomes. Polyploidy is common in plants, but not in animals. Polyploidy are more normal in appearance than aneuploidy. Okay, deletion, duplication, inversion, all of this. Let me talk about it on Monday. That's the end of this chapter. Yes, that's the end of this chapter. But Wednesday, let's talk about deletion, duplication. That's when, just like mutation, but this is in chromosomes. When the chromosomes are deleted, duplicated, inverted, when one piece of chromosomes goes to another, not crossing over, but translocation. These are all interesting. So I want to talk about it at the beginning of the hour when you're fresh, when you have all of your mind together. That's why I do not want to talk about it now. It's getting toward the end of period, and then I think